Hello everyone and welcome back to Easy Dentistry, a channel that makes dental education incredibly easy and interesting. Bacterial growth curve is a frequently asked question in microbiology exams as SAQ, Vivas as well as MCQs. So, we are uploading this very important topic today. But before we begin, please subscribe to our channel. Bacterial growth curve is a short topic, but it is very important. So let's understand concept of what is bacterial growth curve. So let's begin. When a bacteria is inoculated in a suitable liquid medium and it is incubated, bacterial growth occurs. Now this bacterial growth follows a certain course. If the bacterial growth counting is done at certain time interval, it results in a growth curve. Now this bacterial growth curve can be divided into four phases: the lag phase, the log or the exponential phase, the stationary phase and the phase of decline. So this is the diagrammatic representation or graphical representation of the bacterial growth curve viable count. So it is divided into four phases: the lag, log, stationary and the phase of decline. Now let us study each of this phase in detail. First, the lag phase. The lag phase occurs immediately following the seeding of the culture medium. So this first phase is the lag phase. You can see here, this is the lag phase of the bacterial growth curve. It occurs immediately following the seeding of the culture medium. Now, in the lag phase, no appreciable increase in the cell number occurs, but there is a definite increase in the cell size. Now, in the lag phase, the lag phase is the time it required by the microorganism to adapt to the new environment. So the time required for the microorganism to adapt to the new environment is the lag phase. Now this adaptation is needed for enzymes and metabolite to build up in adequate quantities for the multiplication to proceed. So this results in lag phase, and the duration of the lag phase depends upon the species of the bacteria growth, the size of the inoculum, the nature of the culture medium and certain environmental factors such as temperature. So these are the four factors on which the duration of the lag phase depends. Now the next phase is the log phase, which is also known as the exponential phase. Now after the lag phase occurs the log phase. As you can see here, this ascending straight line is the log phase. Now after the lag phase, next occurs the log phase, in which the cell division occurs and there is increase in the cell number exponentially or by geometric progression. If the log of the viable count is plotted against time, as you can see here, in the log phase you find an ascending straight line. Now next comes the stationary phase. After the log phase comes the stationary phase. You can see here, this is the stationary phase. In the stationary phase, the cell division stops, and this division stops due to depletion of the nutrients from the culture medium and accumulation of toxins in the medium. Now, in the stationary phase, the number of cells which are formed are equal to the number of cells which die. So, the viable count remains stationary. So, it is called as the stationary phase. Also, in the stationary phase, there exists an equilibrium between the dying cells and the newly formed cells. Now last is the phase of decline. So you can see here in the viable count there is a phase of decline. In the phase of decline, the population of the cell decreases due to cell death. Now this cell death occurs due to three factors: exhaustion of the nutrients, toxin accumulation, and accumulation of autolytic enzymes in the culture medium. Now if we consider the total count and not the viable count, and if graph is plotted for the total count of the bacteria, there is no phase of decline. So you can see here, this is another graph for the total count. The viable count, this is the lower graph which shows lag phase, log phase, stationary phase and phase of decline, while the graph of the total count do not show any phase of decline. So for total count there is no phase of decline. But you should know here an important point, that in case the bacteria are autolytic, the total growth shows the phase of decline too. So if the bacteria grown are autolytic, then this phase of decline do exist for the total count too. Now, let us now consider some of the important points to remember and which are frequently asked from the topic bacterial growth curve. Now, the maximum cell size is attained towards the end of the lag phase. Secondly, small and uniform cell staining occurs in the log phase. Gram variable staining, that is gram positive or gram negative staining, and irregular staining due to presence of intracellular storage granules occurs during the stationary phase. Sporulation, production of exotoxins and antibiotics occur in the stationary phase, while involution forms of the bacteria are seen in the phase of decline. So you should remember these points because these are the frequently asked questions in MCQs. Thanks for watching this lecture, and if you understand the lecture, please subscribe to our channel and hit on the bell icon if you want more such lectures on microbiology. That's all for now. Happy studying.